A lot of things. Yeah. So Danai Kontou is a PhD candidate at the University of Cambridge in the Department of Ecology and Evolution. And Danai began her academic career at the University of Patras in Western Greece in the Peloponnese region of Greece. I believe I got that correct. Where she also worked as a research assistant, working with fossilized animal bones. After obtaining her BS in biology, she worked in a sea turtle management company called Archelon. I've actually heard of this company, also based in Greece. And in 2018, Danai obtained her research master's from the University of College London in biodiversity, evolution, and conservation. After this, she worked as a graduate research assistant on the genetic structure of UK kelp populations. So we're bouncing around a lot in terms of taxonomy here. And in 2020, Danai joined the lab of Dr. Andrew Tanit-Zap, sorry, at the University of Cambridge, where she researches how Daphnia, preserved in sediment, respond to changes in acidification and to invasive species in Canada. Danai's research has been published in Insect Molecular Ecology and PLOS Biology. And with that, I will let Danai present her talk today entitled Tales from the Deep, Tracking the Evolution of Aquatic Food Webs in a Changing World. And I apologize for the pronunciation on my part.
No problem. Hi, everyone, and thank you very much, Rob, for the kind introduction. Let me start by sharing my screen. It worked well before. Hopefully, it will work again. Can we
Great.
see the slides?
Yep, looks great.
You can see the proper one, correct? Like,
And it's correct, just the slide and nothing else.
okay, perfect.
Mm-hmm.
Fantastic. Okay, so again, thank you very much for the introduction. Hello to everyone. I've already spotted a number of Daphnia people in the audience, which is slightly intimidating, very exciting though. And I want to start by saying that I'm actually in the last year of my PhD, which feels slightly terrifying saying out loud. I got the SSE award a couple of years ago now, and it very generously funded the sequencing costs for the experiments that you're going to see later in my talk. So first of all, not working. Let me see. No. Yeah, there you go. Okay, so I usually describe myself as an evolutionary ecologist. I come from two very different backgrounds. I've always been very interested in evolutionary biology, but I'm also an ecologist at heart. I have a background in conservation, so I like bringing the two fields together to see how answering evolutionary questions can help us solve environmental problems. So in this talk, actually, it's sort of split into two halves. I want to give you an overview of the methods that I've used in my PhD. And I want to start by talking a little bit about Canadian freshwaters and sort of the bigger picture, giving you some necessary ecological context to understand the questions that I'm working on, and then hopefully convince you that zooplankton is a great model system for study evolutionary biology, and then going a little bit more in depth into the exact methods and the natural experimental setup that I am exploiting for my PhD. And then finally, I'm going to present the results from the first two chapters of my PhD that I'm currently working on submitting very soon, and a bit of a sneak peek of the third chapter that I'm working on and hoping to complete in the next few months, and then the predictions that I have for future steps and what Daphnia is going to be doing. So to start, I don't believe that I have to stress the importance of having access to clean and safe fresh water, or how essential fresh water is for life on the planet. However, a question that I get very often is why Canada of all places? Why they just wake up one morning and decide to work there? Well, Canada has about 62% of global lakes that are larger than 0.1 square kilometer, that is permanent lakes, and an impressive 20% of global freshwater reserves are in Canada. So all in all, a very good place to be if you're a freshwater ecologist and you're at all interested in freshwaters. Now, in the last couple of decades, freshwater lakes in Canada and across the Northern Hemisphere and elsewhere on the planet have been facing a series of extreme environmental pressures. They're extremely valuable, but also very vulnerable to a combination of stressors. This is just highlighting a number of them. A lot of the lakes in Canada have faced acidification that led to catastrophic calcium loss, and are further threatened by global warming, salinization, eutrophication in some areas, and also biological invasions that are greatly facilitated by the changes in climate and human activity. Now, when we studying each of those stressors in isolation is a bit of a simplistic view on reality, because what actually happens in natural systems is that a lot of those stressors interact with each other. And that means that it's not often easy to separate their effects. And I want to highlight acidification and biological invasions in particular, because in my study system in Canada, I'm focusing on the interplay between acidification and a very, very successful biological invasion. And I'll talk to you more about it in a second. So that was a bit of the kind of ecological background and context and then significance. But because we're at the Society for the Study of Evolution, what are the sort of evolutionary questions that I'm interested in and how they click together? So the two big questions that I'm interested in are about the predictability of evolution. So does evolution repeat itself under similar environmental conditions? And how long does it take for a wild population to change? I am by no means the first person to think about it, wonder about it, try to work on it. And there's some brilliant research out there that I'm not even going to begin to try and compare my PhD to. However, most of the existing research on the debates about contingency versus determinism in evolution roughly follows two main approaches. The first one is what we call parallel evolution experiments. There's some very famous examples on bacteria, like Professor Lenski's long-term experiments, and also brilliant work in Drosophila and plants. And what those experiments do is that we start with an ancestral population, very similar population or even identical, and then we monitor those populations through time to check for signs of divergence or convergence under a control environment. And the second type of experiment, which is what we commonly find in field studies, is what we call historical evolution experiments. In this case, we will start with populations that are different, that have independent trajectories, and suddenly find themselves under similar environmental conditions. And we want to see how they've responded, how similar the responses are. And there's some, again, very well-known long-term monitoring studies, just a couple of examples. The work of Jonathan Losses et al. In the Caribbean on anolis lizards, the grants, finches in the Galapagos, and a lot of lovely work on sticklebacks as well. Both of these approaches have their benefits, but also come with certain biases and limitations that can be either the initial selection of the artificial selection of ancestral strains, and also in the case of historical experiments, very often we have to, historical evolution experiments, very often we have to rely on certain inferences about the past, especially in the case of vertebrates, because there is a very hard limit on how far back we can actually go. So we base our hypothesis on contemporary data. So ideally, in a perfect scenario, we want to have the same genetic pool that we can track and manipulate through time and see how it's been responding to change in a natural setting. And why do we care so much about predictions? Predictability of evolution, the predictability of evolution is a very interesting question in evolutionary biology and genetics, but it's a crucial one in ecology and conservation, because accurate predictions are what allow us to make informed decisions that can help us with managing vulnerable ecosystems and can ultimately assist in our conservation efforts, which are becoming more and more important as climate continues to change rapidly. And to give you some more specific context about the region I work in, I was very lucky to work in a beautiful region, the beautiful region of Muskoka, in the Halliburton Highlands in south central Ontario, just a couple hours north of Toronto, for those of you that are familiar with their area. And these are the these are the 12 lakes that you can see here in the picture, and they all share the same environmental history, let's say. So this area is actually very popular now. It's a very popular tourist region. It's got about 1,600 lakes and mostly temperate climate. It used to be densely forested. It still is to an extent, and relatively inaccessible. And the troubles start around the mid-1800s when the first European settlers arrived in the region. Now, this region is on top of the Canadian Shield, and that means that the soil is not very suitable for agriculture, so it was never exploited as such. Instead, the main industrial activity was logging and mining. This was greatly facilitated and intensified in the early 1900s when the railroad expanded all the way from Toronto towards Muskoka. And the railroad expansion also meant that the area was more accessible and became more popular for tourists at the time. So we started having more visitors, steamboat cruises, cottages being built, holiday homes, and also around the 1920s and 1930s are the earliest records that I managed to find of artificial stocking of the lakes with popular fish species such as bass and trout for recreational fishing. With increased popularity and increased population in the region, the provincial government started actively monitoring the lakes in the 1970s, recording annual changes in biodiversity, shoreline development, and water chemistry. And that is precisely how we know that the lakes acidified due to past industrial activity all the way from the 70s to the 80s, which resulted to a catastrophic loss of calcium. And although there was an effort to reverse the damage, and the lakes have mostly recovered in present day, unfortunately, the calcium that was lost was never really replenished. On top of that, in the early 2000s, a lot of these lakes were reported as invaded by the Eurasian spiny water flea, and I will tell you more about that in a minute because it's the main focus of my PhD. And today, the main threats around those lakes are increased shoreline development. More and more holiday homes are being built, more roads, wires, more people. And with that comes another threat, which is salinization. Because there's more roads and more houses, people pour salt for ice, and all that ends up in the waterways and spring. And there's actually a big project going on at the moment in the region studying the effects of salinization in those lakes. And moving on to my model organism and model system, as I said, I saw some familiar Daphnia faces in the audience. But for those of you who are not familiar with this little creature, Daphnia polycaria is a very common species in those Canadian lakes. It's a large-bodied microcrustacean. And a keystone grazer that feeds on the algae and keeps the waters clear. It is an excellent model species, same as the other 100 species of Daphnia, and it's been characterized as an ecogenomic model. They're very responsive to environmental change. There's a wealth of genomic data because they're very easy to handle in their laboratory, and they're transparent, so people have done all sorts of interesting experiments with morphology, physiology, behavior, but perhaps their most fascinating feature is their unique life cycle. Daphnia polycaria in particular reproduces with what is known as cyclical parthenogenesis. And if that sounds like a bit of a mouthful, what it means in reality is that it alternates between an asexual and sexual phase every single year. So they're all female when the conditions are great in the lakes, especially during the summer and spring where there's enough food available. They just reproduce asexually, they clone themselves. and produce daughters. But when conditions get worse, and with the winter, Daphnia polycaria in those North American lakes produces males. There is sexual reproduction, and that results into the production of large diploid embryos that are protected within this chitinous structure called an ephipium, and the ephipia are then released when the adult female molts, and they end up eventually in the sediment. And that creates a very nice layered record for species that do that annually, such as Daphnia polycaria. And the ephipia are actually, especially from Daphnia polycaria, actually quite large. If you isolate them from the sediment, you can actually see them with the naked eye. You don't even need the microscope. And what's really nice is that to an extent, we can also tell the species. You can see here a variety of ephipia that I isolated from the sediments of my lakes. And you can see the differences in size and the differences in shape. Because they are crustaceans and they have to molt, the ephipium grows within the cavity of the female. So the bigger the female, the bigger the ephipium, and also it assumes a broadly species-specific shape. Imagine like baking a cake to a specific mold. And at the bottom, you can see in the white circle there, you can actually see a transparent ephipium, because one of my lakes, they're usually melanized to protect the embryos, but I actually found a lake where most of the ephipia were transparent. And you can clearly see one of the embryos on the left, the green one, that's still in the blastula stage, and the other one that started to look a little bit more like an individual. You can see the eye and the top, that little black dot, and you can see that it's pink as well. Usually one of them will hatch, and they can stay preserved in sediment for hundreds of years and only hatch with when there's suitable environmental cues. The other species that I'm working on is Bithytryphis. So Bithytryphis is also a cladoceran. Is actually a cousin, let's say, to Daphnia, but instead of feeding on phytoplankton, it actually preys on Daphnia, and it can remove up to 30% of the native zooplankton biomass in lakes where it's been introduced. Originally, it's native to large clear-water Eurasian lakes. It is a gape-limited predator, so it will go after prey that's smaller in size. And it's got this very characteristic spine, this barbed spine that is preserved in sediment because it's also made of chitin. And that also makes it relatively unpalatable to young fish. Which creates a problem because it can very quickly explode in population density. It seems to benefit from warmer temperatures. It was allegedly first detected in the late 1980s in the Great Lakes, starting from Lake Ontario, and we know that spread is greatly facilitated by human activity. And these are just examples of signs that I found everywhere in lakes, basically requesting people to take care when moving between lakes to reduce the spread. And it can very, very easily spread with fishing. There's been some really good research showing that it's the #1 way with which it's spread, actually. And what happens when the spiny water flea invades those lakes? Just a simplified version of a food web in the lakes. Daphnia keep algae in check. They're much smaller. This is kind of to give you an idea of the scale. They're much smaller than Bithytryphis. So when Bithytryphis is introduced, very quickly decimates native populations. That means that algae has the opportunity to explode, and that can have catastrophic consequences for the wider lake community. And just to drive the point home a little bit more, Daphnias are important because they can keep our waters looking like the left and not the right. It's been estimated that US states have spent about $1.5 billion in the last decade alone dealing with harmful algal blooms, not just because of Bithytryphis, but also because of climate change. So once again, very important to make sure that those food webs are healthy so they can sustain the freshwater communities and the ecosystem services that they provide. And I'm now going to move into the methodological part of the talk. The thing I've enjoyed most about my PhD is trying the field of paleoecology, which allow us to travel back in time. Lakes are excellent natural archives. Their sediments can preserve a lot of information. And to do that, we just have to dive beneath the surface. A very quick overview of what is usually done with sediment cores, and what I've done for my PhD as well, is we can extract cores from its lake. Those cores can then be dated with isotopes such as 210, lead-210. And then we can use the profile that we get from the isotope dating to get those nice slices that correspond to specific time points. And then from those time points and sections, we can then extract a number of proxies. We can have microfossils, we can have diatoms, we can have environmental DNA, very often entire organisms, such as Daphnia ephipia, and also do chemical reconstructions, such as the pH and the calcium from the diatoms, chlorophyll, carbon, phosphorus, and a number of other proxies. And in order to get the microfossils and the ephipia in particular, what I've done was filter sections of sediment, and then those filtrates can be very easily observed under a microscope. It looks a little bit like this. This is actually a very nice example. There's loads of ephipia, and I've just highlighted a few here for you. This is basically what I've spent the last 3 years of my life staring at. Slides microscope plates that look a little bit like this. And this is a very nice example because it's relatively easy to play with the contrast and pick out the ephipia. A lot of the samples, unfortunately, look a little bit like this. And yes, there are ephipia in there, highlighted. There you go. After we've extracted and isolated the ephipia, though, we can go one step further, and that is with resurrection ecology. And before the Jurassic Park soundtrack starts playing in your heads, I'll have to stop you there. It is a very exciting field, but probably not as exciting as having a T-Rex roaming around. It's not really what we do with Daphnia, but it's close. Essentially, we can hatch Daphnia individuals after hundreds of years, and we can use them to experiment and see how they compare to modern populations. And I just have here 2 examples from a lake in Minnesota. Those are 2 of my favorite papers that I read very early on in my PhD and was really excited about. And I think they're a very nice example of what you can do with resurrected Daphnia. So in the first case, the researchers resurrected compared 3 different clones, that they called modern, recent, and ancient, and did thermal tolerance assays. They basically checked how well they respond to increased temperatures, and they found that contemporary clones actually exhibit higher thermal tolerance. And then in another paper, the researchers looked into core profiles looked into eutrophication and phosphorus retention, and they also found that contemporary clones are better at retaining nutrients when competing against ancestral clones. And the 2 clones, the ancestral and the modern clones, have different growth rates and different phosphorus concentrations. And what was really exciting from this particular paper as well is that the researchers also included a population structure analysis. And for anyone who's not familiar with a structure plot, basically each bar is an individual and each color represents a specific genotypic cluster. And then the proportion of the colors within the bar gives you the probability of a certain individual belonging to one cluster or the other. And you can clearly see that there's a change here from red to blue. And you'll be seeing some more of those later on from my results as well. So there's been evidence of rapid evolution that's been documented in wild Daphnia in response to major stressors, and these are another two papers that I'm very fond of. One also looks at Bifidobacterium longimanus, but doesn't include any genomic data. But more recent studies, more and more studies include genomic data, usually from polls of clonal lineages that have been established in the lab. What's really nice, though, is that we can also sequence directly from the resting eggs without the need to resurrect. And in previous work done by Luisa Orsini, that is just as representative. So they said that they proved that it is just as representative as looking into a clone that you've resurrected and established in the lab, and just 5 resting embryos can accurately represent a specific time point. And this is just something that I also validated in the first year of my PhD with a model lake, where I just quickly scanned a pool of resting eggs for 22 microsites, and I looked at observed, expected, and expected heterozygosity rates and how they changed. And basically, I got the plot that you see on the slide, which was identical to what Luisa Orsini and et al. Had reported in their own research. So, and we see a similar situation for allelic richness as well. And the problem with sequencing eggs directly is that there is a trade-off where it is a lot trickier to do and you get lower genome coverage. But when you're trying to do a PhD with very limited time and resources, it actually helps you by not having to wait a lot for individuals that might or might not hatch. And also it can potentially reduce the bias, what we call propagule bias, which is basically the non-random selection of ancestral genotype when you sequence only the things that manage to hatch after 100 years. So the main questions that I've been trying to address for my PhD, there were a lot more when I started, kind of more innocently thinking that this would be a lot easier to do and I could work on a lot more things. But eventually I had to limit myself to those 3 key questions and kind of going back to the bigger questions that I presented in the beginning. So in those 12 lakes that I am studying in Canada that share a similar ecological history, can we observe a repeatable response in Daphnia when faced with a common stressor? And how does temporal genetic structure of Daphnia differ among similar lakes? And the other question was, how long has the spiny water flea been present in North American lakes? And that question is going to make a lot more sense in just a bit. So for my PhD, what I have is a set of 12 lakes, 8 invaded and 4 control. This is all thanks to data, long-term monitoring data that I have from the Ministry of the Environment. I've got 68 cores in total that record about 200 years of change in the region, although I'm mainly focusing on the last century. I have water chemistry data for these lakes. I've got about 2,000 plus sediment filters that have been processed and 170 good resting egg genomes and about 8,000 measurements from complete, perfectly preserved FEPM from across my lakes. So the first thing was to check how comparable my sites were. So as I said, I wanted sites that had similar environmental histories and comparable characteristics. This is where they are on the map. And it was also important that they all had data and that I could pick invaded and true controls, lakes that where Bifidobacterium has never been detected. And I also checked the sediment to make sure that we have absolutely no evidence. So I started with a bigger set of lakes where data was available from the Ministry of the Environment. You can see here that the darker blue lakes are the ones that are invaded by Bifidobacterium. The control lakes are in green on the other side. And you can see that there is a separation between the two, which was why it was really important that I checked my lakes and selected suitable controls that were comparable in chemistry. So I could then answer questions about the effects of predation and not just differences in populations because of differences in water chemistry. So after I did that, I selected my 12 sites that were accessible. And while I was sampling them, I also took water samples, and I got those tested for chemistry, for chemical parameters as well, just to make sure again that there were no significant differences between invaded and uninvaded. We only noticed a very small difference in total phosphorus, but it was really small, and everything else was identical. And it's such a small difference that we don't consider it to be very biologically relevant for those particular lakes, at least. And this is also some more data of what the lakes looked like back in, well, I'd say present, but back in 2021 when I sampled them. This is what what the surface of the water looked like and what the pH. You can see that the pH is going back to normal in most cases. However, again, I need to remind you that the calcium stayed very, very low. And what we do with the sediments after we've collected and filtered, you end up with something that looks a bit like this. And the first step was to date as well. I'm not going to bore you with loads of isotope graphs, but I'm just going to give you one example here of one invaded and one uninvaded to explain how we establish timelines and how we exclude problematic cores. So in my case, I was lucky. All of the cores had this really nice profile, and by really nice profile is, I mean, a curve like the one you see here in pink. We want to see a nice steady curve, a nice curve, and a nice steady decline as we go further down the core in our isotope profile, in our lead profile. Any weird up and downs mean that there's probably mixing in the core, so we can't necessarily trust the sections we're looking at. And that can happen due to handling at the field, or just natural causes such as bioturbation. And it's important to do that and to align the profiles correctly, because different lakes are going to have different sedimentation rates. So we want to make sure that when we're comparing sections, we don't just go by depth, because a 6 centimeters in one lake might not represent the same time as 6 centimeters in the other lake. So it's really important that when I extract populations from Daphne populations from those sections, I can align them properly. And this is another example of the sedimentation rates and the dating profile. And the reason that the dates stop to a point is because 210 PB is accurate only up to 150 years. If we're going to go deeper, we need to use different isotopes. And but in my case, since I was focusing mainly in the last 100, PB was absolutely fine. And for the eggs, those were extracted, identified, measured, and dissected one by one. I sorted the eggs by looking at viable embryos, like ones that looked a little bit like A and B, that were in good condition. I could potentially get some DNA out of. I did a lot of trials in my first year with embryos from various stages of preservation to see how much DNA we could actually get. And after we've got our embryos, it's just a matter of genomic DNA extraction and library preparation, and then whole genome sequencing, for which the SSE very generously provided funding. And what have I found? So I've tried to track the abundance of both Daphnia and Bifidobacterium through time. Ephipia are predictably more abundant in sediment from the last 3 decades due to natural preservation, but there are some clear trends in density. You can see, for example, Fletcher Lake in orange. The colored bars are the Ephipia, black bars are the Bifidobacterium spines. You can see that there is some fluctuation over the years, and that's more clear in lakes such as Crown and Fletcher, where we have a really nice record going all the way down. The only exception was Solitaire, which was originally reported as invaded by Bifidobacterium, but I didn't find any evidence of Bifidobacterium, and I asked around and no one else had found. So although it is listed as an invaded, in the end, I end up including it as a control. And the other important trend to note is the recovery from acidification. So the green lines that you see here are the reconstructed chlorophyll A profiles from the sediment cores. The brown lines are the dissolved organic carbon. And although these are just, this is just depth and doesn't show the dates, I want you to pay attention to this dip that exists in all the profiles. This dip corresponds to the salaces between 1970s and 1980s at the time of the peak acidification in its lake, and you can see how the lakes start to recover somewhat. This was just to check where my acidification layer is and what was the overall trend. However, an unexpected result that came from this is the following. So the dust line is where I was expecting to find Bithytryphis spines in the red sediment record, which is a little bit deeper than the layer that would correspond to the date of first report, because we know that Bithytryphis can actually exist in those lakes a little bit earlier, up to a decade before it reaches a density that's enough for it to be detected with conventional net sampling methods from the water. But actually, what I found was that Bithytryphis appears way deeper in the sediment, which was very unexpected. And I decided to look into it a bit more because I wanted to compare populations of Daphnia before and after the introduction of Bithytryphis. So when Bithytryphis actually appears in the record, it was very important for me to establish this kind of event horizon of before and after. And this is an example of Leech Lake, lake that I'm using as a case study to see what actually happened with the invasion of Bithytryphis, to reconstruct the timeline of the invasion. We do detect spines way deeper, but the ones that are the bottom sections are about 200% like lower in density. But the peak of the spines in the record coincides with the time of first detection. And I've also detected Bithytryphis mitochondrial DNA from qPCR with qPCR from bulk sediment DNA from the same course, along with spines. So that record agrees with the spines. The peak that you see here highlighted in the dashed boxes is consistent amplification of a Bithytryphis specific of a CO1 fragment with Bithytryphis specific primers that were designed by Walsh et al. For a different study where they were trying to track Bithytryphis in eDNA samples from the water. So I tested the same in sediment and was very consistent across multiple lakes, never appeared in the controls, and I saw it in multiple replicates for its sediment sample. So more evidence that it was probably there a bit earlier than anticipated. And finally, perhaps the most exciting bit is that we, thanks to a collaboration with NERT and Queen's University, we actually managed to date Bithytryphis microfossils with radiocarbon, which has never been done before. That is very challenging due to the small sample sizes and young age. However, we tested how we collect the number of cores. We tested how well they correlate to each other by reconstructing the chlorophyll A profiles and dating them. And then we pooled individuals across multiple cores, and we then had enough material to actually test the samples with radiocarbon, and that was done thanks to SWERC, the NERC facility, ISTO facility up in Glasgow. And what they helped us do is trace the 20th century carbon peak with Bithytryphis samples, which was the main challenge when dating with radiocarbon samples that are very young in age. And we also tested some plant materials for calibration. And what we have so far is that dating pretty much confirms that Bithytryphis was present in the region decades earlier than originally reported. And that's probably the result I'm most excited about in terms of my PhD. And we are preparing this manuscript, so watch this space. But the next question that I asked myself was, does it make any sense for Bithytryphis to be there so much earlier if conditions weren't perfect? If we put the two, if we combine all the data into like this one nice plot, we do see sort of a pattern emerging. So you can see how the FVP pretty much follow the nutrient availability and the the lakes, the chlorophyll and the DOC, and the recovery. And the interesting thing, though, is that Bifidobacterium appears right before the acidification peak, which could explain why it managed to explode and establish itself, because it's in its native range, it likes clear water lakes. So the lakes acidify, the water clears, Bifidobacterium has the opportunity to expand, and also at the same time it finds very vulnerable prey, because Daphne is a calcium-rich species that has already been made vulnerable by the calcium loss following acidification. And a way that it could have been introduced potentially is through artificial fish stocking, is my current theory. But of course, this needs to be looked into further. And the other idea is warmer temperatures. We know that Bifidobacterium benefits from warmer temperatures, even in its native range as well. And the squares that I've highlighted here actually show some periods of high summer temperatures in the district of Muskoka, and they actually match the first square. The first period is the 1930s, 1950s, and that matches the time where I'm finding it in the sediment. And also you can see those two peaks further on to the right that correspond to the late 1980s, when Bifidobacterium was first detected in the Great Lakes, and then the early 2000s when Bifidobacterium was first detected, again by net sampling, meaning very, very high densities in the water in the region of Muskoka. So those are my current theories. And what about Daphnia? So we've seen a significant increase in minifipia length, which is a proxy to also estimate maternal body size in the last 100 years. So when comparing populations from the bottom sections of the course, they right before Bifidobacterium appears in the sediment, or at the onset of Bifidobacterium, when the first couple spines appear, to the top modern populations from 2021, when I sampled, we see a significant increase in size. This was highly significant in invaded lakes and much smaller in uninvaded lakes. So as you can see here, the increase in size is a consistent response across all lakes, but it's more than double in the ones that have been invaded by Bifidobacterium. And if we expand the dates a bit further and actually include all of the, not just the tops and bottoms, but also the in-between slices, to look at the trends over time, going from way before the industrial activity started in the region all the way to modern times, we still see this kind of same consistent increase. And I'm currently playing with models to sort of examine the combined effects of acidification, predation, and nutrient availability that came with recovery from acidification. And I am again seeing the same pattern of an increase in size across lakes, and its combined significant effect of both predation by Bifidobacterium and the increase in chlorophyll A concentrations. And when comparing the genotypes of Daphnia, this is a PCOA of the genotypes between the tops, and of the same genotypes between the tops and bottoms of the lake. They're just centroids for its subpopulation. I wanted to see if there was a pattern. I was initially expecting to see big differences between the tops and the bottoms. In some cases, we do see that tops are away from the bottoms within the same lakes, but actually, in most lakes, top and bottom populations are relatively close. Instead, the pattern that I saw emerging was a geographical one. So I saw that actually lakes that were further apart in the landscape and the modern day genotypes were more different than lakes that were further apart, and I tested that with a simple Mantel test, which makes sense. And when looking at population structure comparisons between the top and bottom, I can see that different clones dominate the top versus bottom sections in the invaded lakes. This is a structure plot, same principle as the one I described before. Its bar is an individual resting embryo that has been sequenced, and its shade of color represents a specific genetic cluster. We found significant differences in lakes that were invaded by Bifidobacterium, at least in 4 out of the out of the 6. There are a few lakes missing here, and that's because for some of the lakes, I didn't manage to recover enough good quality eggs to have genotypic data. So although the complete data set looking at the size and morphology is 12 lakes, it is slightly smaller for genotypic data, because I tried to be quite strict with the quality of the sequences. And I selected another a specific subset of lakes to also look into population structure a bit more in depth. So I actually sequenced individual layers as well that also cover the period of acidification, and that's what I'm currently working on. Try to see where the effects of acidification start and where the effects of predation start, because when we include those middle layers, you can see that suddenly there are some changing in the control lakes as well. So that is the kind of project that I'm focused on at the moment. And going back to the top versus bottom assay, the nucleotide diversity across the genome for all of my Daphnias was low and maintained in the last 100 years, especially in the controls. In the invaded lakes, however, we do see those peaks in nucleotide diversity, and it's significantly higher in invaded lakes compared to uninvaded. And the same goes for genome differentiation. In the plot here, I'm only showing FST values that were more or equal, higher than 0. This is a pairwise FST comparison between top and bottom Daphnia subpopulations. Again, very low in the control lakes, but completely different picture in the invaded lakes, where we have high differentiation between top and bottom. And I've highlighted the outlier, the FST outliers that in color, the FST outliers that fall within the top 1% of the genome-wide FST estimate. This is just a very simplistic view of what's going on with outliers, and I'm hoping to explore it a little bit further and more in detail, because the focus of that first chapter was essentially just looking at the size response and the population structure. And this is perhaps a clearer, more condensed view of what's going on with the outliers. Again, the more color there is, the more outliers. They were evenly spread across the genome in most cases, and quite different. And Fletcher and Grandview in particular were 2 lakes where we had a lot of outliers. And my first thought was perhaps there were 2 lakes that had better coverage, but that was not the case actually. And we did check to make sure there were no significant differences in coverage between tops and bottoms and between different lakes that could introduce bias into how many outliers we're seeing. And what about those outliers? So I first looked into regions that have been described as controlling body size for Daphnia magna, specifically 5 QTLs that control most of the body size variation in Daphnia magna. I didn't really see anything going on there, but Daphnia magna is actually very different to Daphnia polycaria, even though they are in the same genus. So my next step was to check 20 loci that I managed to find from studies that have been done on Daphnia pulex and Daphnia polycaria, and that have been associated with body size development and Phylipia production in lab experiments. Out of these, only 4, there were only 4 loci where I found outliers from my own samples, and I identified about 120 potential outlier hotspots from my samples. And I had a quick look at what are the molecular and biological functions. And interestingly enough, I didn't see as much body size, but I did see metabolic pathways, protein modification, cell cycle, DNA repair, and a lot of LNC RNAs, which are really interesting because LNC RNAs control a lot of epigenetic effects and maternal effects, and play a big role in epigenetics and maternal effects in Daphnia. So there's probably something that's going on in there in terms of response to environmental stress. So the next step is hopefully to do a more in-depth and more precise analysis. And I'm going to end with just the kind of most likely answers. Can we, to the questions that I posed earlier, can we observe a repeatable response in Daphnia when faced with a common stressor? The answer is yes. We did see increased nucleotide diversity across all embedded lakes, increased genome differentiation, and a 14% increase in mean FVPS size. And population genetic structure consistently changed following bifidobacterial establishment and severe acidification. And the spiny water flea has probably been around since the 1950s, potentially since the start of the 20th century, and certainly longer than the last 40 years, which has big implications of how we've been studying this system. And I've already said that I am working on a more in-depth analysis to try and disentangle the effects of predation and acidification in those lakes where one starts and when one ends. And obviously, I'm hoping that anything that we identify in those studies would be very helpful for future resurrection experiments, validation with transcriptomics about the exact roles of those any genes identified, and also more eDNA studies. So I really hope I can continue this work, or other people can continue this work. And I think we're close to time now. So I just want to end by saying that you can't do great science without great people. So I just want to quickly acknowledge all the wonderful supervisors, mentors, friends, colleagues, collaborators from both sides of the Atlantic across multiple countries and institutions for making any of this possible in the first place. And of course, all of our funders and collaborators, including the SSE and my college and department from the University of Cambridge that have supported my PhD work through the years. And a big thank you to the SSE for inviting me, and to all of you today. And I'm very happy to answer your questions, because I understand I ran through quite a bit of information very fast. So thank you.
All right, Danai, thank you very much for a great talk. Lots of data for the audience to digest and ask about. We just have a comment here from Dagmar. Sorry, I cannot stay. Many thanks, and look forward to reading the papers. And it looks like Mary Jane has a question here with her hand raised. She can go ahead.
Yeah, of course.
Yes, thank you. I first have to say that it was a really fascinating talk, lots of interesting natural history, especially for people like me who aren't Daphnia people. And I really liked your pointing out that lake sediments are natural archives, and the fact that you could resurrect Daphnia at different levels. I thought that was, and that you exploited that so well. The question I have comes from my own interest in development in relation to evolution, and especially in flexible phenotypes, alternative phenotypes. So that's sort of where this question is coming from. And I wondered, so you did a lot of comparisons, say, with top and bottom Daphnia, the genes, the genetic, the structure, population structure at both levels. And the thing that worried me a little is that I wonder if it's possible that, so you're looking at ancestral and current Daphnia genomes. So you're implying that evolutionary change has happened, which seems likely. But I wondered if the selection that's going on could be at least partly due to the ability to estivate so long, or to have what you're calling a resting phase that happens so long, so hundreds of years. So I can imagine that, or I would hypothesize that there might be selection for long duration individuals, and by the time you sample, say 100 years after a deposit was made, you might be sampling the best survivors. And that could be part of the evolutionary change that you can document. So I wondered what you thought about that. I also, well, that's one question. I have another one, but I mainly would like to hear what there may be evidence against this idea. So that's what I'm wondering about.
So I'm very glad you like the talk, and thanks for the comment. It's actually a really good question. So there's been a little bit of conflicting evidence. So depending on the population, so in some lakes, there's been shown that the resting egg bank actually acts as a bit of a genetic buffer. So what happens is that when some of the ephipia open up, so some of the hatchlings would naturally, some of the embryos will naturally hatch, not artificially, not by resurrection. So they do hatch when they receive the right environmental cues, usually light and temperature. So the population is sort of replenished, and it's why certain studies have suggested that in some cases Daphnia has exceptional temporal stability. So you have the same kind of generalist clones that do really, really well, and you have this buffer in between. So you see this low diversity through time that is maintained. And in my case, I think that we can actually make a comparison but it's better if you have, because everything that I sample, it is a snapshot in time. And I compare it to another snapshot in time, which is whatever was at the top. The other thing that perhaps wasn't very clear from my talk and the way I presented things is that every sediment section is a 2 centimeter section that actually covers more than 1 year. So you do get this kind of nice window, this pool. I just pick random eggs that represent multiple years. So it's not just a single year. That's the other benefit of sampling directly from sediment. So you get this kind of periods of populations of Daphnia. Ideally, I would have liked to have sequenced like the entire core in every single lake. That would have been fantastic, but it wasn't feasible, both in terms of time and money. But I think it's way more informative if we can also sequence the steps in between. This is why I showed some of the lakes where I've actually sequenced in between, the in-between sections as well, because I feel like that gives a more complete story, and we're not just relying on this top and bottom populations where nobody really knows what happened in the middle, as you said. So yes, ideally we would sequence the entire core going down.
Yeah, but it still seems like, I think it's reasonable to compare extremes even. But since some of the differences you noted had to do with metabolite and metabolic characteristics and epigenetic things, which are developmental differences. And so that, I still have the same worry that there could be that selection operating on developmental capacity to last many, many years. But anyway, it's something to think about. And I think your answer is part of the answer. If you see continuous change, for example, in one particular aspect, and you can have ideas about why that changed, that's a good answer.
I believe that's probably the explanation in one of my lakes, for example, Fletcher Lake, where I see a huge difference between the top and bottom. And my suspicion there is that basically whatever clone was dominant whatever clones were dominant at the bottom, where something happened in between and were completely replaced by something else, because it's just so much different when compared to other invaded lakes. But unfortunately, unless there's more experiments and I can actually hatch and do developmental assays and all that, it's a bit difficult to answer these questions. But yes, it is something to think about.
If it turns out that these are alternative phenotypes, in other words, not all Daphnia can, what I would call estivate, have a resting stage, then you could actually have from the very beginning alternative genetically determined differences in the ones that estivate and the ones that don't. So, I think that thinking about the intervening development might help think about this stuff. But thank you.
Thank you.
Yeah, it's a very interesting question. we're over to that would put an extra variable that wasn't to consider into the data. We're over time now, but I had almost an opposite question, which so Dr. West Eberhard's question was like, are you filtering for only the genotypes that can last? And I was thinking, like when you're doing this type of sampling, and I was thinking, well, are there also genotypes in the embryo stage that don't make it to adulthood that would be filtered out in the natural population since you're taking embryos? So sort of like a lack of filtering in that respect, although all samples would be equal there. It's not like the older ones are different than the younger ones there. They all just have embryos. So just sort of a similar question, but yeah, very interesting to consider. And but now we're 5 minutes over, and so I'll let you go, Danai. It was great to learn about your research, and we SSE will be heading to that area, not quite that area of Canada, but this summer for the Montreal meeting. So maybe I'll see you all there. So thanks a lot for everybody, and we'll meet again next week.